right then. Now we're talking about light microscope. I think we did by looking at those structures. Yes, you've seen the part of light microscope, but let me talk about it a bit in details. So it says here, generally light microscope uses lenses to form a magnified image of the specimen. So it uses lenses, but it says here, over the centuries there has been great development in the making of light microscope, which has enhanced its capacity on magnification and resolution. Now magnification means ability to make something bigger, so to say, just makes it bigger. Now, while resolution, on the other hand, is showing details of things. For example, when 2 points are close, very, very close together, and they are almost seen as one, a higher resolution we still show that these things are almost seen as one, but in reality they are not together, they are actually separate. So resolution is like showing you the details of things. Most of the time when you don't have high resolution on the microscope, you will see things that ought to be separate as one because it has low resolution. All right, now as I say, even with the advancement of the light microscope, it is the large and subcellular structures like nuclei, that's part of nucleus, and vacuole that can be visible. This, however, has to be stained. Yes, even for you to see it still, for us to see the plant cell, we have to stain it with iodine. For us to see the animal cell, you have to stain it with methylene blue. So, I mean, that is the limitation, and yet you are only able to see the nucleus and some few structures. We are going to really see that when we get to the second part of this microscopy. So it says here, in other words, light microscope simply makes objects appear bigger, not necessarily showing the details of what they contain. This is very important. Now, this is what we can see from the light microscope: the cell membrane for plant and animal cell, the cytoplasm, obviously, the nucleus, both in animal cell and plant cell. Well, we can see chloroplast, we can see vacuole in the animal cell. Of course, we can also see the cell wall, even though I was not labeled here. These are the things we can see underneath the light microscope. All right, but let's move on. All right, we are still on microscopy. I want to show you the electron microscope. Now, it says due to the limited magnification found in light microscope, there has to be formation of a better magnifying instrument, which gave rise to what? Electron microscope. So the limitations that were present in the light microscope has been improved on in the electron microscope. So it says here, unlike the light microscope, the electron microscope uses beams of electrons, that's collection of electrons like that, rather than light to visualize specimens. So the wavelength of an electron beam is much smaller than that of the visible light, which gives the electron microscope a much higher resolution. So in case you're asked to differentiate between light microscope and electron microscope, we can talk about one of the differences is that one uses lenses as a means of magnification, while electron microscope uses beams of electron. That's what goes through the specimen. It literally goes through the specimen really and shows what the specimen contains, like almost everything there. So resolution then is what? The smallest distance at which two closely positioned small objects can still be seen as separate objects. This is important. Now, when you look at my two fingers I press together, it seems they are like it's one. You can say it is one if you look at just this edge here, that it's one. But if this were to be viewed under a very under an electron microscope that has high resolution, you still see the space in between these two, and because it's not one, it's still two, even though I'm making it look as if it's one. So the point is, high resolution is when you can see things that appear as one as separate, and that's because of the wavelength of electron microscope, so of electron, which electron microscope uses really. I wish I can go into details into that. There are a lot of things to say on that, but you will see that in my A-level classes. So basically here, this is an example of an electron microscope. Now let me say something here. There's a downside to electron microscope, at least to most of them, is that you can only work, by the time you go through the preparation of what it takes to put a specimen here, the specimen becomes dead. So you can't work with a living specimen most of the time on the electron microscope, really. While on the light microscope, you can still work with a living specimen, so to say. This is one of the things, the limitation. Okay, can you see what I'm saying? This is the beam of electron that's going to pass through this specimen. All right. Of course, you see that on the screen and all of those things. It's going to show what the specimen looks like on the screen. All right. So it says here, because of the mechanism of operation of the electron microscope, it is now possible to see tiny subcellular structures such as what? Mitochondria, vesicles, ribosomes, peroxisome. Like, see these structures here, lysosomes, ribosome. Ribosome is actually very, very tiny, really. Microfilaments, centrioles. This centriole is actually found only in animal cells. You can see nuclear pore. What we see under the electron microscope was that it just has a nucleus. So under the light microscope is that it has a nucleus. But here we can actually see that they are actually pores through the light microscope, through the nucleus, so to say. So it says here, it also has enabled us to understand the structure of the nucleus and the cell membrane much better. For example, we now know that this cell membrane is made up of a bilipid layer. It means it has lipids. I mean, this, I don't want to go in there now, but this, the A-level student, we know what I'm talking about, that this has, we now know that you have something like that in your cell membrane, in the cell membrane of the cell. You have some proteins like that. We didn't know that before now. We just thought it's just a sheet, but we now understand that we have protein and lipids forming the cell membrane. So it's actually more complex than the way it is. So that is thanks to electron microscope. All right.